The translational session of BTOG, which I was very pleased to participate in, really showcased some very interesting topics. I think obviously antibody drug conjugates was one, but my colleague Colin Lindsay took us through a great whistle-stop tour of the advances in KRAS mutant non-small cell lung cancer. Specifically, he went through some of the historic data of the different methods by which KRAS was thought to be potentially targetable, some interesting alterations that were thought, to be able to select the KRAS mutant population, an approach where certain drugs that were thought to change synthetic lethality could have been harnessed, but unfortunately didn't show any activity. And then he talks a little bit about the newer KRAS inhibitor small molecules, two in particular, one called Sotorasib and another called Adagrasib. And both of these small molecules are directed against a certain type of KRAS mutation called KRAS G12C and were developed through X-ray crystallography techniques. So he talks a little bit about what are the pros and cons of these agents, generally that they are very well tolerated. They've shown some very good activity in early phase studies. And also we appreciate that KRAS mutations can sometimes happen with second alterations, combinations such as KRAS and STK11, KRAS and KEAP1, KRAS and P53, and how we see that these agents appear to have activity across these different co-alterations as well. He also highlights that there may be some relevance in terms of how we target these going forward, and that there may be differences in the combinations. So really a deep dive into the treatment options and our understanding of the biology of KRAS mutant lung cancer. The second talk by Andrew Furness looked at cellular therapies in non-small cell lung cancer and how this may apply in clinical trials and maybe even in future clinical practice. And Dr. Furness has a PhD and an extensive background in laboratory immunology, and took us through sort of the basic principles of how cellular immunotherapy works, where usually a tissue sample, or in some cases now even a blood sample, we extract the cells that we feel may be responsible for the benefit against these types of cancers. We expand them outside of the body and then we reinfuse them back into the patient. This is something called TIL therapy, tumor infiltrating lymphocytes. He goes through some of the data that underscores this approach in patients with melanoma, and then some of the limited but still interesting data that has been published and presented in non-small cell lung cancer using these TIL type approaches, where you see different immune cell populations change and tumors change in response to this TIL infusion. And really, I think some of the take-home messages from this are that while this may be challenging in the case of patients with lung cancers, and sometimes it may be challenging to get a large amount of tissue or to be able to obtain these tumor-infiltrating lymphocytes, there are newer ways to do this using access through the circulation. And there are a number of early phase studies looking to see whether this may show a benefit in patients with lung cancer or may be feasible in patients with lung cancer. So I think really two very interesting talks, and I'd recommend people logging in and listening, because they will understand a lot about the advances in translational lung cancer from these three talks.